Hello and welcome to another video lecture. In this lecture we are going to discuss about programmed cell death. A cell death which is well regulated, which is programmed in the genetic makeup of the cell, and most surprisingly the cell is actively involved in its own death. Apparently the active involvement of the cell in its own death does not make any sense, because usually in multicellular organisms we have hundreds of metabolic activities, and these metabolic activities are designed to extend or to support the survival of the cell. Therefore, the cell is using it as a source of energy to maintain these metabolic activities. But this programmed cell death is opposite, because the molecular events occurring in this programmed cell death are ultimately leading to the death of the cell, and cell is actively involved in it, that is, it is using its energy sources for this activity. So what is the reason behind this active involvement of the cell in its own death? As you can see that in multicellular organisms, the life of a single cell is not so important as that of the life of an organism. This programmed cell death is very important for this life of multicellular individual. That is, this programmed cell death plays very important role so far as the development of an multicellular organism is concerned. This programmed cell death is very essential for the defense of an organism, and it is also very important to maintain the homeostasis of an individual. For example, we are having thousands of these blood cells produced in bone marrow each day, and same number of cells are getting eliminated through this programmed cell death. And in this way, this programmed cell death is involved in maintaining the constant number of cells within the individual. In case of development, we have many structures which are getting eliminated through this programmed cell death. For example, in metamorphosis of the insects or amphibians, we have many structures like tail that is getting eliminated through this programmed cell death. And in case of human embryos, we have this elimination of this tail, or in this elimination of a second fold between the digits of our hands, and it is because of this programmed cell death that these tissues are being removed and new structures are being formed. If in case of infection, if a cell gets infected by virus or a bacteria, our immune system recognizes this cell, and accordingly this infected cell, it gets eliminated by this programmed cell death, and in this way it checks the spread of bacterial or viral infection. In other case, for example, if the DNA of a cell gets damaged, and there are chances that this cell having this damaged DNA or this mutation can turn into the cancerous cell, and this cell having this damaged DNA is eliminated or killed through this programmed cell death, again it saves the individual from the spread of this cancer. During the developmental stages, we have more than 50% of our neurons get eliminated through this process, so that other neurons form the stable interconnections. And this programmed cell death is very important, but at the level of individual, or at the level of multicellular individual level. And this cell, you can say, through this programmed cell death sacrifices its life for the benefit of multicellular organism. This programmed cell death follows a most common pathway, that is this apoptosis. And in this lecture we are going to discuss what is this apoptosis and what are the molecules involved in this process, and how these molecules interact with each other in different pathways. So let us briefly try to understand what is apoptosis and what are the various morphological changes occurring in this process. In programmed cell death, as I said, this apoptosis is the most common pathway, and this apoptosis term was first introduced by John Kerr, Andrew and Curie in 1972. And this apoptosis refers to sequence of morphological changes occurring in a cell which is going to die through this programmed cell death. That is, sequence of morphological changes occurring in the cell which is going to die through this programmed cell death. That is this apoptosis. That is, this apoptosis refers to the morphological changes occurring in a cell. So here you can see that this is a normal cell. When this normal cell will receive a death signal from within the cytoplasm or from outside of the cytoplasm, in both cases, this cell will respond to that death signal. And accordingly, in this cell, we will be having activation of a sequence of molecular events. And those molecular events will have an effect on its morphology. Those molecular events will have its impact on the structure of this cell. And those structural changes refers to this apoptosis. Now this apoptosis have one molecular base, and then we have this morphological base. And here, as you can see, this in this normal cell we have the centrally located this nucleus, then we have this genetic material, and then we have this plasma membrane. Here I have specifically mentioned this phosphatidylserine, and this phosphatidylserine in normal cells is located in the inner leaflet of the plasma membrane. It is in the normal cells. Why I have mentioned it here? Because it plays very important role in the phagocytosis. So this phosphatidylserine is present in the inner leaflet of the plasma membrane. Now when the cell will receive death signal and it starts this apoptosis process, or you can say this programmed cell death, now there will be some morphological changes. Most prominent morphological changes include this chromatin condensation, that is the chromatin or genetic material of this cell will get condensed. Then we have this DNA fragmentation, that is there will be some DNases, or you can say nucleases. Those nucleases will cleave this DNA between the nucleosomes, resulting in the fragmentation resulting in the formation of the different fragments of this genetic material. And then we have this nucleus gets fragmented, that is we have different fragments of this single. Here we have this single nucleus, but here it will lead to the formation of different fragments. And then also we have this shrinkage of the volume of the cell, that is overall volume of the cell gets reduced, the cell gets shrinked. So it leads to the reduction in the overall volume of the cell. And also we have the detachment of the cell, that is this cell in a tissue gets detached from the neighboring cells. It is having any connections with the neighboring cells, these connections gets detached from this neighboring cell, and this cell shrinks and its volume gets reduced, it is chromatin gets condensed, it is DNA gets fragmented, it is nucleus gets fragmented. So these are morphological changes. Along with it, we have these edges, this smooth cornered edges on this surface of the plasma membrane. You can say these, we are having these smooth corner outgrowths on the surface of this plasma membrane. So these are various morphological changes occurring in this cell undergoing this apoptosis. And here at this stage, when the cell gets condensed or shrinked, it gets fragmented in these apoptotic bodies. And here you can see in this case we have this phosphatidylserine lying inside of this plasma membrane towards the cytoplasm. Once these apoptotic bodies are formed, now we have these phosphatidylserine on the surface of the plasma membrane. This phosphatidylserine flips from the inner leaflet of the membrane to the outer leaflet of the membrane, and this phosphatidylserine is getting now expressed on the surface of the plasma membrane. And this expression of this phosphatidylserine on the surface of the plasma membrane now acts as a signal, and these macrophages recognize this signal and accordingly have receptors on their plasma membrane, and through these receptors present on the macrophages, they interact with these apoptotic bodies and then engulf these or phagocytose these apoptotic bodies. So in this way we have this cell. Through the apoptosis, it undergoes various morphological changes, then its cell gets fragmented into these apoptotic bodies, and these apoptotic bodies are then getting engulfed or phagocytosed by these macrophages. As you can see, there is no leakage of cytoplasmic content in the surroundings. That is, in apoptosis, this whole cell is getting engulfed by these macrophages, or you can say in many cases the contents present in these vesicles is able to get recycled. That is, they are being, these contents or these molecules present in these apoptotic bodies can be recycled, they can be reused. So in apoptosis we do not have any leakage of this cytoplasmic content, and it is very opposite to the necrosis. In necrosis we have leakage of the cytoplasmic content into the surrounding, leading to the inflammation. But here in the apoptosis we have this descent way where the cytoplasm gets fragmented in these apoptotic bodies, and these apoptotic bodies are engulfed or phagocytosed by these macrophages without any leakage, without being cytoplasmic content leaked into the surroundings. So this is the apoptosis. So this apoptosis, or you can say the morphological changes occurring in this cell, these morphological changes have some molecules responsible for it. What are the molecules which are responsible for these morphological changes? So let us try to understand the genes and the molecules responsible for these morphological changes which are occurring in the apoptosis. As I said, this apoptosis refers to the morphological changes at the cellular level, but at the molecular level there are some genes involved in it, there are gene products involved in it, and these gene products, these proteins, interact in specific pathways. Now let us first try to understand what are the genes and what are the gene products which are involved in this whole apoptotic process. In order to investigate the molecules or the genes responsible for this well-regulated apoptosis, C. elegans or Caenorhabditis elegans, it proved to be the best experimental model because it is having fixed number of cells. It is eutelic. We have in nematodes we have this eutelic condition, and in C. elegans we have 1090 cells present in this organism. That is, it starts its life by having these 1090 cells. And among these 1090 cells, 131 cells die during the development. It starts its life with 1090 cells, and during the development, 131 cells die through this programmed cell death. And another aspect associated with these 131 cells is that they are specific. That is, each time it is the same group of 131 cells that are dying through this programmed cell death. And this specificity with this death indicates that this process, this developmental process involving the death of these 131 cells is highly regulated. And during investigation, it was found that there are 3 genes responsible for it. These 3 genes are SID9, SID4 and SID3. CE for cell and D for death. So these are SID9, SID4 and SID3. Among these, this SID9 is having inhibitory role on this apoptosis. That is, it inhibits the cell to enter or to proceed or to die through this apoptosis. And this SID4 is pro-apoptotic. That is, it favors this apoptosis, cell death, programmed cell death, these SID4 and SID3. A functional role of these 3 genes was based on loss of function idea. For example, if the gene is mutated, then observe the loss of particular function, and if there is a loss of function in that organism having a particular gene mutated, then obviously that loss of function, that functional loss is associated with that mutated gene. So here, first they were mutated this SID9, then SID4 and SID3, then observe the loss of function, then observe whether this apoptosis is stimulated or it is inhibited. It was found that when this SID9 was mutated, even normal cells died through this programmed cell death. Or you can say even normal cells die when this SID9 was mutated. It was confirmed that this SID9 is inhibiting the apoptosis. When it is present, it will inhibit. Now when the gene is mutated, now this product, now the gene product is not present in the cells, even normal cells follow this apoptotic pathway. So it was proved that this SID9 is having inhibitory role. It inhibits the apoptosis. And these SID4 and SID3, when these were mutated, these 131 cells did not die through this apoptosis, and it was this confirmed that this SID4 and SID3, they are pro-apoptotic. So these three genes in C. elegans were found to be involved in this high regulation apoptotic process or high regulated programmed cell death. This SID9 is having this inhibitory role on this apoptosis, and this SID4 and SID3, they are having this, they are pro-apoptotic. And it was also found this SID9 exerts its inhibitory effect on SID4. SID9, it inhibits this SID4, not SID3. It inhibits on this SID4, and through this SID4 it inhibits the progression of this apoptosis. And this SID3 was found as an actual effector in this among these three these genes. That is, actually it is the SID3 which is the actual executioner of this apoptotic process. These SID4 and SID3 are on the upstream of this signal cascade. On the sequence of these molecular events, this SID9 is at top level, then we have this SID4, and at the lowest level, at the actual executioner level, we have this SID3. And that is why this SID9, it affects this SID4, and then this SID4 will stimulate this SID3. This is having this pro-apoptotic effect, and this SID3 is the actual gene whose product will actually execute the apoptotic process. So these three genes were found in C. elegans. During further investigation, like in mammals, similar gene products were found in mammals also. That is, we are having a set of proteins which are having almost the same function as that of the SID9, SID4 and SID3. As in case of this mammals, we have this BCL2 family proteins which are having the same function as that of this SID9 this gene product. And in case of this mammals, we are having this APF1 factor. Like SID3, we have caspases in the mammals. So these gene products were also found in the mammals, having almost a similar function as that of these SID3, SID4 and SID9 found in the C. elegans. During the investigation on other animals, like in mammals, we found similar gene products as that of this SID9, SID4 or SID3. For example, in case of mammals, we have this BCL2 family members. The proteins in this BCL2 family, we have these proteins, they have the same functional role as that of this SID9. And here this APAF in case of mammals is having the same function as that of this SID gene product in case of this C. elegans. And similarly, we have these caspases found in mammals which are having this similar function as that of the SID found in the C. elegans. This BCL2 includes the proteins having conserved BH domain, that is BCL homology domain. And in this BCL2 family, we have two types of proteins. One is anti-apoptotic and another is pro-apoptotic. And all the proteins present in this BCL2 family, as I said, is having BH domain, that is BCL2 homology domain. And here this anti-apoptotic includes this BCL2 by itself, and another member is BCLX. This BCL2 and BCLXL, they are anti-apoptotic. This BCL2 and BCLXL, these are having anti-apoptotic activity, and they are having 4 BH domains and 1 transmembrane domain. Here we have this BH4, then we have this BH3, and here we have this BH1, and here it is BH2 domain, and here it is transmembrane domain. It helps to attach it in the mitochondrial membrane. So this protein, these BCL2, BCL is having this anti-apoptotic this activity are multi-BH domain these proteins. They are having 4 BH domains. And here we are having these pro-apoptotic. In this pro-apoptotic we have two. One is multi-BH domain, and then we are having this single BH domain. In case of these pro-apoptotic, we are having this multi-domain backs and back. And here we are having this BH3 domain, and then we are having this BH1, and then we are having this BH2. And here we are also having this transmembrane domain. In these multi-BH domain BCL2 proteins, we are having this transmembrane domain also. This helps us in attaching with the mitochondrial membrane. So in this case, pro-apoptotic BCL2 family, we are having this backs and back. These two proteins favor the apoptosis. And here we have this single BH domain pro-apoptotic BCL2 family member, and this is called as BH3 only. And this is BH3 domain. And this BH3 only is not having any transmembrane domain. So here you have this BCL2 family members, and all these members are sharing this BH domain. That is, they have this specific conserved domains present in these proteins. This is having the anti-apoptotic activity. This is the BCL2 and BCLX, same as that of this SID9. Then we are having this pro-apoptotic multiple domain BCL2 family member. This is the backs and the back. Then we are having this single BH domain, that is this BH3 only domain, and this domain is not having any transmembrane domain. So similar to this SID4, we are having this APF as the pro-apoptotic factor present in the mammals, which is having the same functional role as that of this SID. And here, like SID3 products, in C. elegans we have these caspases in case of these mammals, and these caspases have two specific features. One is that they have cysteine residue at their catalytic site. These enzymes, these proteases have cysteine residue at their catalytic site. And second important aspect is that these caspases will act on their substrate, specifically on the aspartate amino acid towards the C terminal of this polypeptide chain. So they have cysteine residue at their catalytic site, and they will act on the substrate, specifically at the aspartate residue. So these are the caspases. And second aspect is that they are always produced as procaspases. That is, they are produced within the cell in inactive form, and they are being activated only when they are required. And their activation involves a cleavage, usually the cleavage of a special fragment from these procaspases, and then they turn into the caspases, that is in the active form. First is that they have cysteine residue at their catalytic site, at their active site. Second is they act on the substrate, specifically on the aspartate amino acid, and that aspartate should be present towards the C terminal side. And then these caspases are produced within the cell in inactive form, that is as a procaspase, and this procaspase is then turned into the caspase in the active form when it is required. Normally it is present in the inactive form. And in this caspase we have two types of these caspases. Usually they have been divided into two groups. One is the effector group, this effector caspases, and another is these initiator caspases. Initiator caspases usually are the starting caspases. They activate other caspases. In the sequence of these molecular events, these initiator caspases are present at the top level, and they are first activated. These initiator caspases are first activated, and then these initiator caspases activate these effector caspases. And it is this effector caspases which is involved in producing the morphological changes, and actual execution of this apoptosis is through these effector caspases. Here, as you know, in this initiator we are having for example caspase 8 or caspase 9. These are initiator caspases. And here in this effector caspase, for example, we are having this effector caspase 3 or effector caspase 7. So these are two groups of the caspases, and these two caspases, as I said, are proteases which are having this cysteine residue at their catalytic site, and they act on the protein specifically on aspartate residue towards the C terminal, and they are produced in as pro-caspases, that is, they are produced in inactive form. And some caspases are present and at the top level of this molecular eventus, and they initiate this apoptotic process, and they are first stimulated or turned into the active form, and then these initiator caspases activate these effector caspases, and it is this effector caspases actually which produces the morphological changes, or which actually executes the apoptotic process. As I said, these caspases are produced in inactive state, that is as procaspases, and when they are required they are being turned into the active caspases. Once they are in active form, they target almost all proteins. For example, main targets of these caspases include these kinases, protein kinases. For example, this focal adhesion kinase, once it is inactivated, the cell loses its adhesion with the neighboring cells, and thereby it detaches from the neighboring cells by the inactivation of this focal adhesion kinase. Second target of these caspases is these laminas. These laminas, as you know, are the proteins present in the inner nuclear membrane. Once targeted, they cause fragmentation of this nucleus. Third target is this cytoskeleton elements like actin or microtubulus, and once targeted by these caspases, they change the shape. They are involved in the changing shape of this cell during this apoptotic process. Then we are having this caspase activated DNases, and these DNases, as you know, cause fragmentation of this genetic material or this chromatin. So these caspases are the actual executioner of this apoptotic process. So here we are having these genes and their products involved in this apoptotic process. These morphological changes observed during this apoptotic process in a cell are actually caused by these proteins, by these genes. We are having this SED9, SED4 and SED3 in case of C. elegans, and similar products were found in the mammals. We are having this BCL2 family. In this BCL2 family, we are having the proteins which support this apoptosis or which inhibit this apoptosis, which promote this apoptosis or which stimulate this apoptosis or which inhibit this apoptosis. And these BCL2 family members are characterized by these BH domains. We are having this BH3 only domain, then we are having pro-apoptotic this backs and back BCL2 member, and then we are having this anti-apoptotic BCL2X, BCL-XL. They are having this APF protein which is similar in function as that of this SED4. It is pro-apoptotic, and then we are having this SED3. They are called as the caspases, and these caspases are actually which execute this apoptotic process. We are having two groups. One is this initiator, and then we have this effector, and these caspases affect, these caspases target kinases, protein kinases in the cytoplasm of the cell, and these laminins, cytoskeleton, and then we are having this DNases which are being acted by these caspases, and these caspases execute actually the apoptotic effect. So after going through these molecules, let us now try to understand how these molecules interact with each other, or how these molecules stimulate or inhibit each other in the molecular sequence. And accordingly, we are having two pathways in this programmed cell death. One is the extrinsic pathway and second is the intrinsic pathway. The extrinsic pathway, as the signal is coming from outside the cell, then we have some special receptors present on this plasma membrane, and these receptors are called as the death receptors, and this is a family of these receptors which includes some tumor necrosis factor receptor 1, or you can say the fast receptor. So these receptors are present on the plasma membrane which receives the death signal coming from outside of the cell. Here in this case we have this transmembrane receptor which is having this extracytoplasmic domain, then it is having this transmembrane domain, and we are having this cytoplasmic domain. And here on this extracytoplasmic domain, this tumor necrosis factor binds with this tumor necrosis receptor. Tumor necrosis factor, as you know, are the chemicals which are being produced to kill the tumor cells, and here these tumor necrosis factor are trimeric proteins. They get bind with this tumor necrosis factor receptors present on the plasma membrane, and this interaction between the tumor necrosis factor and its receptor is through this extracytoplasmic domain. Once this tumor necrosis factor binds with this extracytoplasmic domain, it causes conformational changes in here in this cytoplasmic domain of this TNFR, and this TNFR cytoplasmic domain recruits other proteins like RIP1 kinase or TRAD or FAD. These proteins get assembled at this site, and all these proteins have common death domain. You can say this receptor is having this death domain towards this cytoplasmic side, and just like that these TRAD or FAD, these are also having this death domain. These are common death domains where through which these different proteins interact and get assembled at this site, and the last protein which assembles at this site is this procaspase 8. This procaspase, as you know, it is a caspase which is in the inactive state, that it is in inactive form at this site, and two molecules of these procaspase get assembled at this site. It is at this site that these procaspases get activated. These two molecules of procaspase which have assembled at this site will now cleave one another, and because of this cleavage these two molecules are activated and they turn into the caspase 8. You can say that at domain of this TNFR present in the cytoplasm, we have these assemblage of multiple proteins, and these multiple proteins assemble at this site through the common death domain, and we are having this procaspase, this initiator caspase, it gets assembled at this protein complex, and then these two molecules of these procaspase get activated and they will turn this procaspase into the caspase 8. Now this initiator caspase is now in the active form, and in this active form this caspase 8 will act on procaspase 3, and this procaspase 3 by the action of this caspase 8 will now get activated and will turn into the caspase 3. And it is this caspase 3 which is the actual executioner, or it is this caspase which actually executes the apoptotic effect. That is, this caspase will now act on these kinases, or it will act on the laminases or cytoskeleton or this GAD. So activity of this is the final caspase which actually executes the apoptotic effect on the cell. This caspase can also interelect with another proteins like BID. This BID belongs to this BCL2 family, that is BH3 only protein, and this BID protein then stimulates the intrinsic pathway. That is, this caspase 8 can either go through directly to activate the procaspase 3, or it can also activate this BID protein, BH3 only protein, and this BID will, through the intermission of this mitochondria, will lead to the activation of this procaspase 3 through the formation of this apoptosome. That is, you can say that the distinction between the extrinsic and intrinsic pathway is not sharp, that these two pathways have some interlink with each other. That is, some molecules which are being activated by this extrinsic pathway can also activate the intrinsic pathway. So here in this extrinsic pathway, we have this receptor present on the plasma membrane, and this receptor gets bind with this tumor necrosis factor, and once this ligand binds with this receptor, it causes conformational changes in the cytoplasmic domain of this receptor. This conformational change then recruits other protein complexes like TRAD or FAD. This is TRAD is the TNFR receptor associated dead domain, or we have this FOS associated dead domain. So these protein complexes along with this rib cage, these get assembled at this site, and then they stimulate or then they activate this procaspase into the caspase form, active form, and this caspase then acts on this procaspase 3, and it leads to activation of this procaspase into the caspase 3, and this caspase is the actual executioner of this apoptotic effect. This caspase 3 acts on these protein complexes present within the cytoplasm. So this is the extrinsic pathway of this programmed cell death, and in extrinsic pathway we are having the signal coming from the external source, and from the receptors this signal is being transmitted down into in this series, and finally it reaches to the this caspase 3, which is the actual executioner of this apoptotic effect. Another pathway is the intrinsic pathway, and in intrinsic pathway usually we have the signal coming from within the cytoplasm, and let us try to understand the molecular events in the intrinsic pathway, and what are the various molecules which interact with each other in this intrinsic pathway, and they finally lead to the activation of these effector caspases, this caspase 3 or caspase 7. And let us try to understand what is the intrinsic pathway. Now compared to extrinsic pathway of the programmed cell death, in intrinsic pathway the signal is produced within the cytoplasm of the cell, and many factors can stimulate this intrinsic pathway. For example, increase in calcium ion concentration, or stress caused by the deficiency of the oxygen, or in case of any DNA damage. And in intrinsic pathway, most important role is played by the BCL2 family members. That is, these proteins play very dominant role in the intrinsic pathway. As I said earlier, this BCL2 family members, these include these anti-apoptotic proteins. Then we are having two types of these pro-apoptotic proteins. One is having this multi-domain, multi-BH domain, this pro-apoptotic protein is like BAX and BAG. Then we are having this BH3, that is single BH domain, this BH3 only proteins. In this we are having this BID, BAD or PUMA or Noxa. So these are BCL2 proteins, and these play very important role in the intrinsic pathway. For example, if we are having any kind of DNA damage, if this is DNA and this DNA gets damaged, in a DNA damage we have increased concentration of P53 protein. When is the P53 concentration increases, this P53 concentration stimulates this BH3 only proteins. Normally in the cells we have low expression or low activity of these proteins, BH3 only proteins. These are the main determinants, or these are the main proteins of this BCL2 family which regulate the apoptosis through the intrinsic pathway. These proteins, these BH3 proteins will affect both this pro-apoptotic BCL2 proteins, and they will also affect this anti-apoptotic BCL2 proteins. That is, they can affect this apoptosis by two ways. One way is that they can stimulate these pro-apoptotic BCL2 proteins, and this normal cell can turn into the apoptotic cell. And another way is that they can inhibit the anti-apoptotic activity of these BCL2 or BCL1s.
the anti-apoptotic activity of this BCL2 is inhibited, the cell will enter into the apoptosis. That main determinants of this apoptosis through the intrinsic pathway are these BH3 only proteins. These can be bid, bad, or this puma. So this one is this P53 concentration gets increased. One is this expression of this P53 gets increased within the cytoplasm. It affects the expression of these BH3 only proteins. That it will stimulate, that is, it will cause the expression of the gene encoding these BH3 only proteins. Once these proteins are increased in the cytoplasm, or their activity is increased, they will stimulate this back. So here you can say that this increase in P53 concentration will now have its effect on BH3 only proteins. Here it will effect on this, that is P53 will increase the expression of these BH3 only proteins. Once this BH3 only proteins will increase their activity or expression, they will influence, they will affect the pro-apoptotic BCL2 proteins like BAX. Once stimulated, this BAX will now easily translocate from the cytoplasmic side and will get inserted in the outer mitochondrial membrane here. Along with this BAX, we are also having this BAG protein. As I said earlier, we are having this pro-BCL2 protein is BAX and BAG. This BAG protein is present permanently in the outer mitochondrial membrane, and this BAX is present in the cytoplasm, and this BAX is stimulated by this BH3 only protein. This BH3 only protein can be bid, bad, or puma. These proteins stimulate this BAX and then it translocates into the outer mitochondrial membrane. This BAX protein along with this BAG, they get assembled in the outer mitochondrial membrane, and once assembled they form a channel, or you can say this assemblage of this BAG and BAX in the outer mitochondrial membrane, it increases the permeability of this outer mitochondrial membrane. Once the permeability of this outer mitochondrial membrane gets increased, the content is present within this mitochondria get released from this mitochondria into the cytoplasm, and most important molecule or most important protein complex which gets released through these chambers, through these channels, is this cytochrome C. And this cytochrome C plays very important role in the apoptosis. How? In the cytoplasm, this cytochrome C will assemble with another protein that is called as APAF. You can say that this cytochrome C in the cytoplasm will get assembled with this APAF, and together with this pro-caspase 9, they lead to the formation of this spherical complex that is called as the apoptosome. Now we are having these cytochrome C along with this APF and pro-caspase 9 present in this spherical complex, and this spherical complex, as I said, is called as apoptosome. We are having these 7 subunits arranged in this spherical complex, this apoptosome, and this we are having this cytochrome C, APAF and pro-caspase 9. It is in this complex that this pro-caspase 9 is getting activated, and it leads to the formation of caspase 9. Now this is activated form of pro-caspase 9. Once this pro-caspase is activated and it is formed as a caspase 9, this caspase 9 will act on pro-caspase 3, and this pro-caspase 3 will get activated into the caspase 3. Or you can say this extrinsic pathway or intrinsic pathway both get linked at this caspase 3. In extrinsic pathway, we have the final executioner as this final activation of this procaspase 3, and in intrinsic pathway also we have the activation of this procaspase 3. That is, both these pathways, whether it is the intrinsic pathway or the extrinsic pathway, both these pathways finally execute their effect through these caspases, caspase 3, or it can be the caspase 7 also. So we are having this pathway, this is the intrinsic pathway, and in this intrinsic pathway this mitochondria plays a very dominant role, and along with this mitochondria we have this involvement of these BCL2 family members. In pathway we are having these receptors present on the plasma membrane, and these receptors then lead to the activation of through cytoplasmic domain these procaspases, and they finally activate the procaspase 3. In intrinsic pathway, mitochondrial role along with the BCL2 family members are important for the formation of this apoptosome. This is the complex which activates this procaspase 9, and once this procaspase 9 is activated, it activates then finally this procaspase 3, and this caspase 3 will then attack on these kinins, laminins or cytoskeletons, and that will result in the morphological changes of the cell. So this is about the intrinsic pathway and extrinsic pathway of this apoptosis or programmed cell death.